Good morning, all you Suhai AP Biology students. Rise and shine again. It's 5:45 a.m. and dude, I am stoked to get started on our next topic, enzymes. We've been working our way towards these specialized proteins for a few weeks now, and we're finally there. So get your notes out and something to write with, and let's catalyze. Well, you won't see what I did there because you really don't know what catalyze means, but you're going to learn very quickly, and then when you think about what I just said before, you'll say to yourself, oh, I see what he did there. First, let's talk about the word metabolism. You see it right here, the word metabolism. We're going to use it often in the next few weeks, so let's understand what metabolism really is. Write in your notes that metabolism is the sum, remember the sum is an addition problem, the sum of all of the chemical reactions performed by a living organism. So metabolism is the sum of all of the chemical reactions performed by a living organism. A good example would be photosynthesis in plants, the one that produces food. The other would be cellular respiration, which takes that food and turns it into energy for all living organisms. There are thousands more, but those are two common examples. Now, these are just two examples, but there are thousands of coordinated reactions that have to take place in order for living things to stay, well, living. Recall our discussion of energy flow over the summer in our conservation and ecology units. In this process now, do you remember this process and its efficiency level? Remember that only 10% of the energy is passed from one level to the next in any food pyramid or chain, just like you see here in this picture. Now also recall the differences in the way plant and animal cells look and what organelles they contain. Can you identify which is the plant and which is the animal cell and why? Hopefully you chose this one as the animal cell and this one as the plant cell, and that you could list 3 or 4 reasons why you chose what you chose. Obviously this one has a cell wall. That's usually the one that's the giveaway, but it has chloroplasts here and a large vacuole, etc., etc. So again, that's review, but it's things we're going to talk about again here fairly quickly. So the point we want to make, and for you to get in your notes, is that life is built on chemical reactions, which means organisms transforming energy from one form to another. In chemical terms, this process involves breaking the chains that bind organic molecules together. Remember the 4 organics? We just got done with them. Carbohydrates, proteins, nucleic acids, and lipids. Now breaking those bonds releases the energy contained in them, so it can be used to power these chemical reactions that you see here. Now we're also going to revisit ATP, adenosine triphosphate, the energy storage molecule. And remember, ATP has its own cycle. It's adenosine triphosphate, and remember, it stores energy in those 3 phosphates that it has. So when we break off one of the phosphates, ATP becomes ADP, adenosine diphosphate, and from that broken bond we get the energy. Okay, remember that? So again, we're going to revisit ATP in our discussion now of enzymes as well. Okay, so back to metabolism. Remember that this word describes all of the processes or chemical changes in an organism or single cell by which food is built, referred to as anabolism. There's another new term. In that we make that now into what's called living protoplasm. You might want to write that down. That might be on an exam. Protoplasm is the beginning form of life. Now, so anabolisms are synthesis reactions. They're building molecules. We call those anabolisms. We're going to talk about anabolic reactions. Just like you've heard something called anabolic steroids, we're going to talk about those as anabolic reactions. Now we're also going to talk about the opposite, which is the molecules that break bonds. Those now, understand our hydrolysis reactions, and we'll talk a little bit more about that review-wise here in a second. And digestion would be an example. They're not anabolic reactions. Those are catabolisms or catabolic reactions. So you might want to get those two in your notes. Anabolic reactions build molecules. Dehydration synthesis is an example. Catabolic reactions now would break the bonds between molecules. And hydrolysis is the way that that's accomplished. And digestion would be a great example. Now let's look at some very general examples of what it is enzymes do for these chemical reactions. I'd like you to get each of these diagrams in your notes right now. Now, what I also want you to make sure, so look at the top now, we have a dehydration synthesis, and we have a hydrolysis, so get these reactions in your notes. And also, don't forget to include these things, enzymes. All right, so make sure you label the enzyme in your diagram as well. Now we can clearly see that the reaction on top is a dehydration synthesis, meaning the bonding of monomers together to make a larger polymer. Here's our polymer, here's our two monomers. Now we know from previous discussions that one of these monomers donates a hydrogen ion, while the other donates a hydroxide we call a hydroxyl group. When you join these two together, you form water. An H and an OH forms this H2O. So water is given off, which is why we call it a dehydration synthesis. Now also remember that we've now given this synthesis reaction a name, a metabolic name. It's now called an anabolism or anabolic reaction, a building type of reaction. So in metabolic terms, this is an anabolism. In the bottom, we can see just the opposite, hydrolysis reaction. That actually requires a water molecule in order to break up or digest a polymer into its monomers. In metabolic terms, we now call this reaction a catabolism or catabolic reaction. In your notes, write that the enzyme's job is to speed up this process, mainly so the organism can conserve the energy required to perform this reaction. We call this maximizing reaction efficiency. So make sure you get that in your notes. So these labeled reactions need to get in your notes, and then you need to understand that the difference between an anabolism up here and a catabolism down here. And then here is an actual example. The reaction on top is an anabolic dehydration synthesis, where a molecule of sucrose, all right, here's a molecule of sucrose, is built from the monomers glucose and fructose. And a water molecule now is given off. Now, do you see that the glucose donates the hydroxyl right here? And that the fructose now donates the hydrogen ion. Then in the bottom, we see the catabolic hydrolysis, or breaking up of that polymer into these two monomers. So kind of crisscross now, we see now the difference between the two. You also need to see that a water molecule is required to form now the hydroxyl group in glucose and the hydrogen ion now in fructose. Remember that in chemical terms, this makes it a Bronsted-Lowry acid. So the fructose, because it's a hydrogen acceptor, write that it becomes a Bronsted-Lowry acid. And now because glucose now accepts the hydroxyl group, it makes it a Bronsted-Lowry base. I taught you that if you had me in chemistry, and I'm sure Mr. Wicks and Mrs. Dale taught you that as well. But it's good to get that back in your notes now because you may see that as a question on an AP exam. So remember, a hydrogen ion acceptor is a Bronsted-Lowry acid, and a hydroxyl or hydroxide acceptor would be a Bronsted-Lowry base. And again now, we see the enzyme now in here, and remember the enzyme's job is to maximize the efficiency of both of these, and we'll see how that happens very soon. But I want you to get in your head that it's a catalyst, and catalyst's job now are to maximize the efficiency of a reaction, and we'll see how that happens very shortly. Okay, some more review for you. Write in your notes that exergonic reactions, you see now the word exergonic reactions, give off energy from the breaking of bonds. They release energy. Examples include the digestion or breaking up now of polymers like we just saw on the previous slide, and we now realize that these would be catabolic reactions like hydrolysis. The breaking down now would be a catabolism or a catabolic reaction. You should also understand that by breaking up these molecules, we increase the entropy now. Entropy by making these molecules more random or less organized. Remember that entropy is the measure of the randomness now of a system. When we break up molecules, the system gets less organized. All right, if it's less organized now, it increases now its entropy. Please write that in your notes. Now also write in your notes that in endergonic reactions, here's endergonic now, not exergonic, these are endergonic reactions. Energy is required in order to make them happen. Examples include anabolic reactions that build or synthesize, like dehydration synthesis. Okay? So these would be anabolic or an anabolism type thing. Again, you should also understand that we're actually making these molecules less random now. By making them less random and more organized, we're reducing their entropy. And we're going to talk about it in terms of higher and lower energy states here very, very shortly. Okay, so the last thing we'll do in this podcast is get these two graphs in your notes. You'll be seeing them a lot in the next few days and weeks. And make sure you label each one correctly. Notice now that the y-axis is split up into a top and bottom section. Now, this top section says energy supplied, and the bottom section says energy released. So make sure you get those broken up correctly. Now, it's important that you label them correctly because it's where enzymes will provide the biggest advantage. Okay, now let's talk about the G in this diagram. You see the G right here, and this delta means change. All right, so this would be a negative change in G. This would be a positive change in G is how that would be written. Now, the G represents something called Gibbs free energy. Get that in your notes. Gibbs, G-I-B-B-S, just like the guy from NCIS. Gibbs free energy. Now write in your notes that Gibbs free energy is the energy associated with a chemical reaction that can be used to do work. So make sure you get that in your notes. Now, exergonic reactions release Gibbs free energy. Therefore, chemists have given it a negative value because this reaction is losing energy. So it gives it a negative G because it's given off. Energy is given off or released. Here it says right here, energy is released. So whenever a reaction produces energy, remember we used cellular respiration is an example. It produces energy in the form of ATP. It's called a negative change in G. So make sure you get that in your notes. In contrast, endergonic reactions require energy. So chemists give them a positive value, has a positive change in G. And a good example of this is the anabolic reaction photosynthesis. It requires a constant source of light energy in order to continue. So make sure you label these things correctly. Also, I'd put the examples in your notes as well. Now, lastly, let's look at the energy spike in both examples. This is what I'm talking about right here. This energy spike. You see it here, and you see it here. Now, write in your notes and label on your diagram that this represents activation energy. So right here, put an arrow and label this as activation energy. And then in your notes, write that activation energy is the energy actually required to start a reaction. So we always, in chemistry, use the matchstick as an example. A matchstick is pure potential energy, but you have to strike it in order to release that chemical energy. So once you strike it, you use that heat of friction to strike it, that energy starts, and now you have the exergonic energy because you feel the heat release and see the light, etc. Okay, so that energy required to start a reaction is called activation energy, and we're going to find out that that's where enzymes actually provide the biggest advantage to living organisms. Okay, take a picture of those notes and submit them to Moodle. Grab that study guide, answer the questions, and then I'll see you in part 2 of our discussion of enzymes. Bye.